I'm Cassidy.
And I'm Jenna.
And we're here to teach you about
the Golgi bodies.
First things first. The Golgi is an organelle that is present in all eukaryotic cells, including both plant and animal cells.
They're a very important part of the cell for many reasons, specifically their role in the development of proteins within the cells.
In short, the Golgi bodies act as a warehouse, processing, sorting, shipping, and sometimes manufacturing proteins that come to them from the endoplasmic reticulum.
Some people describe their appearance as a stack of pita bread. Others say that they look more like pancakes.
Although these descriptions sound pretty tasty, this isn't exactly what they are.
We'll start by explaining what the pockets really are and the other parts that make up this magnificent organelle.
There are two different sides of the Golgi apparatus, the cis face and the trans face, and they each serve different functions. They act respectively as the shipping and receiving departments of the organelle.
The cis face receives the products from the ER.
The trans face allows vesicles to pinch off of the Golgi membrane and travel to the destination that needs them.
The cisternae are flattened sacs made up of cell membranes. The membranes separate its internal space from the cytosol within the cell.
After proteins are formed in the ribosomes, which are on the rough endoplasmic reticulum, they are sent in vesicles, which act like cars, to the next phase of their journey. Besides transporting to and from the Golgi apparatus, the vesicles also carry the biological molecules to different organelles within the cell.
As the vesicle approaches the Golgi, the proteins inside have no idea what's coming for them.
Due to the Golgi's cell membrane components, vesicles, which are also surrounded by a membrane, are able to fuse through tubes. It is because of the structure of the membrane in the Golgi that allows this fusion and diffusion to occur.
The proteins enter the Golgi from the cis side and make their way through the organelle until exiting on the trans side.
As they fuse and diffuse through the different layers of the membrane, chemical reactions occur. And as they make their way through the cisternae, they also trade amino acids in order to create whatever protein the organism needs.
As the proteins make their way into the trans side, they begin to sort themselves like the post office ships off packages.
They are preparing themselves to be sent off to the next organelle within the cell, or to the plasma membrane to exit out of one cell and join another one that needs it more.
In summary, as the molecules make their transit from the cis region to the trans region, they are in some way modified.
This whole process is very similar to that of a car wash.
The car enters looking a certain way and exits looking much better after being all fixed up, just like what happens to proteins.
Along with trading amino acids to change proteins, the Golgi apparatus has another function.
It can also create macromolecules from scratch, for example, polysaccharides that are secreted by the cell.
Anyway, you may be wondering, once the proteins are ready to go, how do the vesicles know where to take them?
During the packaging process, different molecules are given ID tags, just like the ones that we put on dog collars in case they get lost. Just as dog IDs show where the dog is supposed to be, cell IDs show where the vesicles should be sent.
An example of a molecular ID could be a phosphate group on its membrane. Yep, the one we learned just last week. PO4.
This will tell the vesicle exactly where to ship its products.
Hey Jenna, are you ready to review?
Let's do it!
So can you tell me where the vesicles come from before they fuse into the Golgi bodies and what their role is within the cell?
That's easy. Vesicles are like cars that transport proteins from the rough endoplasmic reticulum to the membranes of the Golgi. Cassidy, what are the different sections of the Golgi apparatus and how does a vesicle travel through them?
Due to the cell membranes that are part of both the Golgi and the vesicles, the vesicles are able to fuse through the pieces of pita bread and make their way from the cis face to the trans face. I kind of forgot though. What happens to them when they're in the empty space in the middle of each membrane?
You should know this, but I'll let it slide this time. Remember how the proteins come in looking one way and come out looking all tidied up? While passing through, amino acids are added and removed in order to alter the structure and the function.
Oh yeah, I remember now. And once they're all fixed up, they're packaged and shipped off to their next destination, right?
That's exactly right. And it's a nice way to end our explanation of the Golgi bodies.
Thanks for listening, and we hope you learned something new.